My work is really at the intersection of computer science and biochemistry and biology. My lab is interested in developing new technologies to measure metabolism and specifically to discover new metabolites, and then applying those to large-scale data sets from patient samples in order to produce new insights into cancer metabolism and biology. Our work really has two main components. The first is developing new computational tools that can work with large-scale metabolic datasets and link those datasets to the chemical structures of as-of-yet undiscovered small molecules. And the second thing that we do is we analyze large-scale datasets, often from human patients or from mouse models, to actually understand what is the function of these novel small molecules in physiological processes and how is that function perturbed in cancer. The challenge is that we know that there are hundreds of thousands of different small molecules in the human body, but we really have barely scratched the surface of what these small molecules actually are and do. Remarkably, from a computational perspective, we actually have an analytical technology called mass spectrometry that routinely measures tens of thousands of small molecules in any given sample. But the challenge is really in identifying these small molecules from mass spectrometry data. So we are developing machine learning approaches that leverage our understanding of human metabolic biochemistry that's accumulated over decades of study, as well as the data embedded within mass spectrometry datasets in order to translate this data into the chemical structures of as-of-yet undiscovered small molecules that are present in the human body. I really think that this is one of the central challenges of biochemistry in the 21st century. One direction that I'm extremely excited about revolves around a technique called imaging mass spectrometry. And this is an analytical technique that allows one to visualize the localization of every metabolite in the context of a tissue. So for example, you can visualize the localization of thousands of metabolites within a tumor or in the adjacent normal healthy tissue. And we're starting to develop what I feel are some very exciting machine learning approaches that can allow us to really make sense of this data at scale and potentially even identify previously unknown metabolites in the context of the cytoarchitecture of a tumor. Another avenue that I'm very excited about is leveraging large-scale genome sequencing datasets, both from healthy individuals and from individuals who developed cancer, in order to intersect the genetic risk factors that predispose someone to developing a particular type of cancer with either known or unknown metabolites, and ultimately perhaps even discover totally unknown metabolites that modulate a person's risk of developing cancer during their lifetime. We've already discovered more than a dozen new human metabolites that we've been able to experimentally confirm in the lab. So I think that's extremely exciting and really a good sign for what's to come in the future. We are envisioning a future in which we can comprehensively measure and discover all of the unknown metabolic principles that guide the biochemistry within the human body. And I think if we could do that, I'm very hopeful that we can leverage that understanding to better understand cancer biology and ultimately to impact on patient care.